Welcome, everyone. We're delighted to have you join us for today's webinar with Dyne Therapeutics, who provide an update on the registrational expansion cohort of the DELIVER trial of rostudirsen. This is the company's registrational trial in Duchenne patients with mutations amenable to skipping exon 51. This is a very exciting time as the company shares this new long-term clinical data and prepares to file with the FDA for accelerated approval in the second quarter of 2026. I'm Michael Kelly. I'm the Chief Scientific Officer at Cure Duchenne, and I'm going to get us started with a bit of housekeeping. Firstly, you have an opportunity for Q&A today. You can simply click on the button at the bottom of the screen and submit a question anytime throughout the presentation. There'll be a moderated Q&A session at the end of the webinar, where I'll be joined by my colleague, Dr. Liana Orlando, our Vice President of Research. And for those questions that we can't get to, we'll work with the Dyne team to follow up with you after the webinar. And after today, this webinar will be available on Cure Duchenne's website as a resource for the community. So let me begin. Dyne has developed the FORCE platform to efficiently deliver therapeutic cargo in a targeted manner to muscle as well as the brain. This is powerful technology. It utilizes a transferrin receptor binding Fab conjugated to an exon skipping oligonucleotide, efficiently delivering the exon skipping drug to muscle to target the genetic root cause of the disease. To talk more about the platform and provide an update on results from the DELIVER registrational expansion study, we've Dr. Doug Kerr, the Chief Medical Officer at Dyne. So without further ado, let me hand over to Doug to get us started from here.
All right. Good morning, good afternoon, everybody. I am honored to be here today and talking to you about rostudirsen in DMD. I'd like to tell you a little bit about Dyne and what we're doing. Before I get into that, I'd like to just introduce myself. I am a neurologist and a neuroscientist. I spent about 15 years as a professor at Johns Hopkins, taking care of patients with Duchenne and other neuromuscular disorders. So I've had a lot of experience in this space, and I spent the last 20 years or so up in the Cambridge, Boston area where Dyne is, really trying to develop new therapies for neuromuscular diseases, including Duchenne muscular dystrophy. And what I'm going to share with you today is the result of many, many years of hard and excellent work. I do want to call out somebody before I begin, because Maria Naylor, who is on this call, has been here really since the beginning of this Duchenne program and the clinical trial, has done an amazing job and deserves an awful lot of credit for it. So thank you, Maria, for all that you have done. Okay, so let me get into it. Dyne is really about, if we could just go back one, I would just want to hit on that, on a mission to deliver functional improvement for individuals, families, and communities. I just wanted to pause on that because it's really, really important to us. We are using biomarkers, but we really want the therapies that we are testing in clinical trials to result in functional improvement. And I'll show you that indeed that seems to be the case with rostudirsen in Duchenne muscular dystrophy. So we are really, really excited to be able to share these data with you today. So now if we go to the next slide, you are all very much aware of DMD. We have been studying here exon 51 skip amenable DMD. So it is a subpopulation of DMD. It represents about 13% of the entire DMD population. And exon 51 DMD boys often have very severe disease because they do not result at baseline with any meaningful dystrophin in their muscles. And therefore, the muscles are fragile and get progressively weaker over time. So in a sense, this form of DMD is the toughest of the subtypes of DMD that results in progressive weakness of the lower extremities, then the upper extremities, and breathing, as well as cardiac issues. There aren't any great therapies at this point. This is why Dyne got into this space. And I'm going to show you how we developed the FORCE platform to overcome some of the limitations in those therapies that are out there already. So we could go to that next slide. It's okay. So this is a cartoon, but I want to take a minute and introduce you to the FORCE platform. And the Dyne was created to really solve delivery of genetic medicines into tissues that matter in patients and individuals with neuromuscular diseases. The way we do that is in the red and blue. There you can see that's called a Fab, or a fragment of an antibody. It is a part of an antibody that binds to a receptor on the surface of cells in many tissues in the body. That then can bring the genetic payload, which is this antisense oligonucleotide that can induce skipping of dystrophin, into cells. So the top part is really the delivery vehicle. Then you've got a linker, and on the bottom is the genetic medicine itself that can induce skipping of dystrophin. Why that matters is because when you just deliver the genetic medicine alone, it is very, very hard to get enough of that into the tissues that matter. And so we've developed the FORCE platform to overcome that. And importantly, the FORCE platform delivers genetic medicines for the tissues that you see on the right, including muscle. And muscle is skeletal muscle, smooth muscle, as well as cardiac muscle, because we know that cardiac issues are very important in Duchenne muscular dystrophy, as well as the brain. And the brain also matters a lot in Duchenne muscular dystrophy. So being able to deliver the genetic medicine to all of those tissues, we had thought, and now really have very good evidence, that is able to result in functional improvement in Duchenne muscular dystrophy. Okay, next slide. And so I'm going to go here about the trial design. It's called the DELIVER study. And this study was designed and executed by Maria from the beginning to encompass a broad representative patient population of DMD boys with mutations amenable to exon 51 skipping. And that just means what we're doing is skipping the mutation, allowing production of near full-length dystrophin that can better stabilize the muscle. The ages here were 4 to 16 years old and included individuals who were ambulatory and non-ambulatory. And then you'll see that what we did in the bottom is we moved to a registration expansion cohort, which you'll see written here as REC. But that just means after the multiple ascending dose portion of the study, where we were looking at safety, but also the optimal dose and the optimal dose regimen, we then locked that dose and dose regimen in at 20 milligrams per kilogram every 4 weeks and initiated a registration expansion cohort of 32 additional patients. And it is these data that I will show you today that form part of the basis for seeking accelerated approval in the United States. The primary endpoints were change from baseline in dystrophin protein as defined by Western blot at 6 months, along with safety and tolerability. And we also wanted to look at functional change over time. And I'll go over those functional endpoints in a few slides. Just to complete on this slide, in DELIVER, patients had their placebo-controlled period, whether they were in the multiple ascending dose or the registrational expansion cohort, and then have gone on rostudirsen at that go-forward dose of 20 milligrams per kilogram in the open label portions of that study, which are now over 3 years and counting in total. Okay, so we've got we're having a technical problem here on the slide advance, but that's okay. Bear with me here. So we're first going to look at the demographics and baseline participant characteristics. Now, this is a very busy slide. I'm not going to go through each aspect of it. I will orient you to the columns. You can see the gray column is placebo, 24 patients across the MAD and the REC. And then right next to it in the dark blue is the registration expansion cohort patients exposed to rostudirsen. That's 24 patients. And then the last two columns are MAD, multiple ascending dose cohorts, one of which started at 10 milligrams per kilogram, but then dose escalated. And the last one on the right is the 20 milligram per kilogram Q4 week cohort from the MAD. And what we look at in these tables are the baseline characteristics in age, in other medications that people have been on, age of symptom onset, and the bottom of this table is that baseline functional capability. What could these boys do in terms of various outcome measures that will be assessed throughout the trial? So just a few points before we go to the next slide. If you look at the age, across the entirety of the study, the mean age, the average here is 6 to 8.2 years, pretty balanced across the cohorts, but obviously there are patients who were younger and older than this in each of those cohorts. Patients have typically been on corticosteroids coming into the trial and then stably have maintained on corticosteroids through the clinical trial. If you look at prior DMD therapy, some patients have been on other medicines such as eteplirsen or other investigational therapies. And then if you just look at the bottom, it's how they did on these various functional outcome measures, only to say here that they are balanced across groups. And that's very important for us because if there's an imbalance at baseline, it makes interpretation of the results more complicated. And that isn't the case here because these patients are balanced across those groups. Okay, so now we'll look at safety. And so let me spend a minute or two on this because it's a very important aspect of each clinical trial is to understand the safety aspects of the drug. And so we're doing that here. And if you look at the left of the two tables, what you're looking at are TEAEs, which is just a fancy name for adverse events, symptoms that patients have upon entering the trial. treatment emergent adverse events. And you can see on that left-sided table during the placebo control period, which is 6 months, there are some treatment emergent adverse events, but I'll draw your attention to serious related TEAEs, which means obviously a serious event that the investigator felt was due to the drug rather than something that was part of Duchenne, the disease. There aren't any serious related TEAEs, and we were very comforted by that. But if you go a little bit to the right of that, it's a more expansive view of safety. So this includes all study periods, not just the placebo-controlled, but the open label extensions that go out to 36 months. Now, in this, there were 4 patients who had serious related TEAEs. 2 of them we reported last summer, and they were at a higher dose of the medicine than we have been given for the rest of DELIVER and the dose that we will submit for approval. So 2 of those serious related TEAEs were at a higher dose, not explored subsequently. So then what were the 2 patients who had serious related TEAEs? Essentially, they had events of fever and malaise, feeling down and kind of sick around the time of infusions. And the investigator brought those 2 patients into the hospital, considered whether there was an infection, and ultimately discharged those patients. No infection was found. They continue in DELIVER. They did not miss a dose of drug, and they've been continuing to get 20 milligrams per kilogram every 4 weeks since. So it is kind of a infusion-related reaction, or maybe a little bit delayed because this happened 2 to 6 days after the drug was given. And so it's something to watch out for. It happens with most biologic medicines that are infused. And so we feel very good about the safety profile of rostudirsen. Now, if you go to the bottom, it just gives you a sense of why we feel comfortable about the safety profile. And part of it is because we've given so many doses, 1,441 doses, reflective of 113 patient years of follow-up out to 36 months on rostudirsen. And over 1,000 of those doses are at that go-forward dose of 20 milligrams per kilogram every 4 weeks. One more thing before I leave this slide. No participants have persistent related anemia, which is a drop in hemoglobin, which could cause fatigue or other issues. We haven't seen it. And so we're very pleased by that. Okay, so now we'll look at dystrophin. And so what you're looking at here is the change from baseline in dystrophin in the REC. Remember, that's the registration expansion cohort. And so you're seeing that here, 24 patients got drug, and I'll just draw your attention to the low blue bar. That's 0.83% of normal dystrophin at baseline. Very, very low levels of muscle content adjusted dystrophin. And then when those patients get rostudirsen and a muscle biopsy from the bicep muscle is taken 6 months later, the muscle content adjusted dystrophin has increased to 5.46%. And in clinical trials, we look at the p-value. How significant was that? How impactful was it? And you can see the p-value there of less than 0.0001. So very robust, very meaningful, much better than have been demonstrated by other therapies in exon 51 DMD boys. Next slide will be to introduce the 6 functional outcome measures that we will show to you. And so you're familiar with many or all of these, but let me walk through them just very quickly. TTR velocity stands for time to rise velocity, asking a boy to rise from the floor and calculating how fast that occurs. 10-meter walk run velocity below that. The North Star Ambulatory Assessment, which is a 17-item scale measuring ambulatory function. And then if you go to the right of this slide, stride velocity 95th centile is done with an accelerometer placed around the participant's ankle to see how comfortable in their own home, in their own environment, are they walking, and how does that correspond to the speed with which they walk. And SV95C is the 5% of the fastest strides. Pull 2.0 is different in the sense that it is not interrogating or looking at lower extremity function, but upper extremity function, because that really, really matters in DMD. And finally, forced vital capacity as a measure of lung function. And the way these data will be shown is 6 slides, 1 per each of these, and we'll look at the impact on drug versus placebo out to 6 months. So here's the first one. So here is the 10-meter walk, no, time to rise velocity. And so the way to look at this is as you go up from baseline, that means the patient is getting faster. So the velocity is getting higher. And that's what happens on rostudirsen, whereas the placebo patients in gray got slower over that 6-month period. And if you look at the difference between those two, that difference in velocity of 0.04 rises per second exceeds what the Duchenne muscular dystrophy field has concluded is an MCID, which stands for minimal clinically important difference. So that is relevant to patients to have a difference of at least 0.04 on this measure on time to rise. And the p-value here is less than 0.05, which by convention is a meaningful p-value statistical test applied here. Next slide is the 10-meter walk run. And here you see the same thing. Again, going up from baseline over the 6-month period means that the participant on drug is getting faster, whereas the placebo patients are getting slower. That also is right around this minimal clinically important difference. It also has a nominal p-value of less than 0.05, which we think is important. So that's 2. So let's go to the third. And so here is the North Star. Higher scores are better going up from baseline on drug on the blue line, whereas the placebo patients are about the same or a little bit worse. And that difference between those two also approaches the MCID. So trends on all three of those favoring patients on drug. And now here's the fourth, which is stride velocity. And you'll see that both groups go up here. Now, those who were on rostudirsen went up more than those on placebo, but we were a little bit surprised that the placebo group here would also increase. And we think there might have been an outlier where a placebo patient actually did much better than expected. We don't know exactly why that is, but we think that that is probably an outlier. And so we're still pleased that patients on drug are getting faster in their own home, which really gets to a real world improvement in function, which we think is really remarkable. So there's 4, and number 5 is going to be the performance upper limb. And again, going up is better, means more functionality. And we see that patients on drug go up more than those on placebo. So very encouraging. And then we'll go to the forced vital capacity, which is 6. And here what you see is the blue line says, what is the change in the pulmonary capacity over that 6-month period? And those patients on drug were completely stabilized. No decline in forced vital capacity over that 6-month period, whereas patients who were on placebo for that 6-month period declined in terms of their pulmonary function. Now, you might ask, why didn't those patients get better in terms of their pulmonary function? Well, it turns out that they came in with a baseline very high level of forced vital capacity, about 90% predicted. And so there wasn't really much room to go above 90%. But what still makes this really important is that you want to prevent the decline in pulmonary function that happens over time. And we did on drug. And then one point, as before we go to the next slide, is that the placebo-controlled period was only for 6 months. After that period, everybody has access to active rostudirsen. And so we felt that was very important. What we needed to do is some placebo-controlled period, because that's the way we show that the drug works, but we didn't want it to be too long and to provide everybody who came into this trial and sacrificed time and effort and muscle biopsies the opportunity to have active drug as well. But what was amazing about those data there are that we looked at 6 functional outcome measures, and all 6 of them favored drug. Now, those data that you saw were only the 6-month period from the registration expansion cohort, with the gray group being pooled placebo from the rec and the MAD. But what about longer-term data? What about those patients who have been in the study for over 18 or 24 months? and came in during the multiple ascending dose portion. So we wanted to look at those patients as well. So let me orient you briefly to this slide. The column on the left is the original 20 milligram per kilogram cohort from the multiple ascending dose. And what you're looking at is where are those patients 18 months after being on drug. And the way to look at the column below that is you see the vertical line, which is the 0 point. And so if patients are at the 0 point, there has been no change over 18 months. If they're to the right of that vertical line, that means they are better at 18 months than they were at the beginning. And if they were to the left of that line, it means they're worse. Now, we all know that Duchenne muscular dystrophy is a progressive disease. And despite that, if you look at the dots down that left-hand column, every dot is to the right of 0, meaning that that cohort overall is better at 18 months than it was coming into the DELIVER study. Now, it does not mean that every single patient was better. We don't know that yet because we've only looked at this as a cohort level. But it was still amazing to us to see that 18 months later, despite this being a progressive disease, they are stronger, faster, quicker, more upper extremity function, better vital capacity, etc. Okay, so let me now look at the middle of the slide. This is also 18-month data, and it looks at the original 10 milligram per kilogram MAD cohort pooled together with the original 20 mg per kg MAD cohort. So the numbers are a little bit bigger. The numbers here range from 10 to 12, and so it gives you a greater confidence in what these data are telling you. And just like on the left, the patients are to the right of that 0 line, meaning that this larger pooled cohort is functioning better at 18 months than they were at the beginning. And one final time on the right of this slide, this is now 24 months of data. So it's the original 10 mg per kg cohort that got dose escalated to 20 milligrams per kilogram and has been on that go-forward dose of 20 milligrams per kilogram for, I think, 12 to 18 months. And now, 2 years after coming into DELIVER, patients, the dots there are the mean functionality on each of these measures is to the right of 0, substantially so, meaning that even at 24 months, that cohort is better, stronger, faster at 24 months relative to baseline. So that's it for the data that we have at our top line readout. There is more to come, and we're digging into it as well. But there's a lot of things we want to dig into that we haven't had the opportunity to do just yet, but we will. But the conclusion here, really statistically significant and robust increase in dystrophin at 6 months. That is very important because it is required for accelerated approval. And while I'm on that, I just want to say that we don't think that dystrophin is at a steady state or plateau. We actually hope that dystrophin goes up progressively over time. but we needed the 6-month data to support accelerated approval. But then a very good, favorable safety and tolerability profile up to 36 months, functional improvement from baseline observed across multiple clinical measures, and convenient Q4 week dosing. So I will stop there. I'm grateful to have the opportunity. We will use these data and submit to the FDA in Q2, meaning April, May, or June, for accelerated approval. And we are hoping that this drug would then be available to patients in Q1 of 2027, meaning January, February, or March. And now I won't talk about the right side of this slide because it's another of our investigational therapies. And this one, Z-basivarsen, is for myotonic dystrophy. So I will stop there and we can take questions.
Doug, thank you for that. That was quite a tour de force, and I'm glad to see that you won the battle of the slides. You fought with them all the way through.
We did fight them, but that's okay.
Yeah,
Sorry about that.
no worries. Thank you for that. I'm going to invite my colleagues back on again. I think Liana's going to join me for a Q&A session. And as moderator here, I'm going to take the opportunity to sort of have the first few comments about this. First, I'm going to say that the consistency of the data is impressive across multiple endpoints. It's really interesting and impressive and welcome, to see this here drug actually affect quite a bit of muscle system. So congratulations, to you and the team and the data and everyone that participated in this. The
Thank
thing
you.
that struck me the most, I gotta say, was the preservation of lung function at 6 months. I honestly didn't expect to see that happen so quickly. And it really leans into the fact that Z rostudirsen gets into cardiac muscle as well as the brain. And so what we're seeing in lung function probably sort of leans into pretends about what we may see in future for cardiac and particularly following patients for any benefits from the brain. What's the company's view on that there?
We're very excited about what we saw on the forced vital capacity.
Mhm.
Obviously, preservation of lung function, really important. We do know from preclinical work that we've done that we get an awful lot of that medicine to the heart.
Mhm.
Obviously, that is very important to us as well. We don't have any clinical data at this point, although we're hoping to get it to look at what Z-rostudirsen does in the heart. We also have preclinical evidence about accessing the brain. And even in the clinic with our other program, Z-basivarsen for myotonic dystrophy, we've seen some early but very interesting data that you are impacting the central nervous system manifestations of that disease. So it's going to take us some time to sort out really the lung, the heart, the CNS, but we're very encouraged by what we've seen so far.
Me too. And I just got to add to the bottom of that. I'm not expecting to see cardiac effects manifest quickly. It's going to be over a number of years. And that's why I was so surprised and pleased to actually see it come out in the forced vital capacity, because that really leans into what one might expect as we follow this over time.
I agree with you, Michael. That's a very
Yeah.
good point.
Yeah. Liana, do you want to jump in with something?
Sure. We've got a couple of questions submitted beforehand and online during the webinar about steroid use. And so I think I can bundle them together and sort of maybe you can address in the clinical trial why you were controlling for steroids. And what would be the case in a phase 3 trial, how you might address it. And then eventually, whether or not you would have to be on steroids with this drug if it got approved or not.
Yeah, very good questions. And I will tee it up, and maybe Maria will come in and add some additional color to it. But obviously, steroids are important in Duchenne muscular dystrophy. We did not want to exclude patients. In fact, we wanted patients on a stable dose of steroids such that we could look at the impact of Z-rostudirsen without having the confounding variable of a change in steroids. We did not specify exactly what that steroid regimen should be, only that it remains stable. That is going to be the case in the phase 3 study as well. We don't think you need to be on steroids in order to get functional benefit with rostudirsen. But ultimately, I think patients and families want access to whatever they can to help stabilize or halt this disease. And so we felt like being on steroids was important. Maria, what did I miss?
Not much, Doug, not much at all. Again, emphasizing that we wanted to understand the actual effect of our drug on patient outcomes. So that's why we asked that patients be on a stable steroid regimen, and we'll ask the same in phase 3. But agree that the mechanism of action is different from that of steroids. So maybe in a world someday you could envision something else, but for right now, that's what we have in our studies.
Yeah, so just to be specific for some of the questions that come in, you know, if approved, there's no reason why someone who couldn't tolerate steroids wouldn't be able to take this drug.
Correct.
Yeah. And what about switching regimens to something from, you know, like to switch to a Gamry, for example? Um, you know, I think you addressed why that's not possible during the clinical trials, but afterwards?
I think afterwards, absolutely. I think patients and families and doctors need to make the best choice for them. We didn't want that switching necessarily in the clinical trial, but obviously as new therapies and new even steroids, each of these steroids is a little bit different. And if a patient wanted to switch, we would think that would be perfectly acceptable.
Terrific.
Maria, you agree?
I do. We did make an amendment to the protocol to allow for switching later on in the study. So for the first year of the study, we did ask that patients were on the same regimen, really in order to pick up any differences in function. But the idea was at that point, dystrophin is pretty much reached a good, stable, steady state. And so we could envision patients switching at that point. When we started the study as well, Agamree was not approved, and so we did update the protocol to be flexible and up to date with what's going on in the field. We know it's changing very rapidly.
No, that's a really good point, and I think it also speaks to listening to the community and the needs and being flexible where you can. So I'm sure people have appreciated that. For the sort of open label extension and the path towards commercialization, are you going to be continuing those individuals on therapy through all of the regulatory interactions? And what's the timing on the phase 3?
So yes to the first question, patients who entered into Deliver are on drug, will stay on drug. At some point, this becomes available, and they would switch, but we would not want patients to have a dose interruption. So they will be in this until it is available by other mechanisms. And what was the second part? I forget.
The timing of the launch of the phase 3. We're already getting emails from people who are interested in when you're going to be recruiting.
That is going to be in Q2, so April, May, June of 26 is when that study will start.
Yeah, can I just ask, Doug, like I know I heard you talk previously about this. You're feverishly working on completing the inclusion exclusion criteria and really giving guidance on that. Any sense of when the community can learn, what you're looking for?
It's a very important point. We have not done that. We have not given the details yet. We are still ironing those out. We're having engagements with regulators really throughout the world. So we will do that, and it's a very important thing to talk about what the design is of this phase 3. We're getting close, and it's going to be, before we start the study, no doubt, and probably early in 2026 when we do that.
Yeah, I can see that part of the challenge here, you've almost got an embarrassment of riches, in what you've read out here right now. So what are you going to select as primary, secondary? That's really part of the challenge that I see for you.
It is. It is challenging when you've had the kind of breadth of functional improvement, but your input is really critical here. Regulators' input. It's all part of this information gathering. We also look at the statistics and the meaningfulness of various endpoints. So there's a lot that goes into really finalizing that decision because it's so, so important to get that right. And so we'll begin to have these discussions even further with you about the phase 3 as we turn the corner in 26.
Excellent. A couple of comments I'll make, and that's saying you invited comment straight away. The SV95C was interesting to me, and you're quite right. I listened to your comment about, hey, this is real world information of a benefit in there. Not to get lost in that is the delta. I think it was 2.1 on the North Star. Even a single point change in the North Star has got an enormous difference in a patient's lives. It's whether they can do something on their own, whether they can't, whether they need help. And all of this is real world as well. So like I congratulate you on the data, even on the North Star, which I think is very significant for patients and their families.
That's wonderful to hear. Thank you for that.
Leanna?
Yeah, I don't know that you can answer this, but it's always good to hear your thoughts, because there's a question about sort of the mechanism of what you're doing when you're restoring dystrophin. And so do you think that it's slowing the breakdown of muscles, or or could it potentially also be helping with building of new muscles? And what are your thoughts on that?
Yeah, very good question. I mean, I will say that we don't definitively know, but we know the importance of dystrophin in preservation and repair of muscle. And in DMD patients, there's very little dystrophin. And so the muscle cells are very fragile. They start to break down. Some of that muscle is replaced by adipose tissue, fibrosis, and inflammation. And the key thing in our mind is getting that dystrophin expressed. And it's really important that you get that diffusely within the muscle. It's really important that you get that to the heart, to the diaphragm, to the central nervous system. So part of why we think we're seeing this functional improvement is because those tissues are stabilized, not breaking down, and probably repairing, because we know those tissues certainly have the ability to repair. But in untreated Duchenne muscular dystrophy, the repair cannot keep up with the damage that's ongoing. And we hope that we are reversing that process, favoring stabilization and then repair.
Terrific. We've also got some questions here that, since we talked about the phase 3, some more things have come in. So just to confirm, the dose for the phase 3 will be the same for this registrational cohort. So
It
that's
will.
20 milligrams, right?
Every 4 weeks, yes.
Every 4 weeks. Terrific.
20 milligrams per kilogram. Yep.
And is the plan for the phase 3 to be in the US and ex-US? That we have questions about the locations.
Yes, it will be. It will be in the US as well as many other countries outside the US.
Terrific. And then I bet you could have anticipated this question. What are the plans for targeting additional exons?
Yeah, it's of great interest to us. I know it's of great interest to you. We're really excited to explore other exons. We don't want to stop here. We have in our pipeline other exons that we can go after within Duchenne muscular dystrophy. Honestly, we were waiting for these data that you just saw before advancing other DMD programs. But now we have the clinical validation to continue the development of our programs for those other exons. And frankly, we really want to have a discussion with regulators, including the FDA, about a basket trial or a platform designation or ways in which you can expedite the ability of patients with other mutations to get access to these medicines. And part of the reason for that enthusiasm is when we go after other subsets of DMD, that force platform that I showed, it's the same Fab, it's the same antibody fragment, it's the same linker, it's the same chemistry of the genetic medicine. And so we think that that clinical validation should allow you to move more rapidly into and through the clinic with those other exons.
Yeah, I just say that we welcome that. We've been talking about this for quite a while, and the opportunity to use the platform designation gets you into not just the rare exons, but the ultra rare exons. And I think that there really is something that's going to happen over the next few years.
Agree.
Yeah, I completely concur. And just
Mhm.
to, for our audience, like when you talk about a basket trial or a platform trial, and that you want to talk to the regulators about, it's the idea, right, that you could, instead of doing a separate trial for each exon that you're targeting, you actually can do one and have them run simultaneously rather than sort of sequentially.
That's exactly right.
Yeah, so the idea there that you get additional statistical power, I think, from having them go together, and then families don't have to wait as long
Mhm.
for you to move through one after another.
And shared cohorts for placebo.
Yep.
Yeah.
Exactly.
Anything else, Liana?
Um, No, I think we just about covered it.
Okay.
Despite having all of the technical difficulty with the slides, you have a very clear presentation, and I can just see from what's come in that the community is just as excited as we are about the results and eager to learn more when you dig into
Mhm.
the data a little bit more.
Okay, so let me just wrap things up then. So thank you everybody for joining our Shame webinar today. And a big thank you to Doug and the whole team at Dyne for the work they're doing in developing the rostudirsen and for sharing these updates with the community. Shame was a very early investor in Dyne. They were still preclinical at the time. And you can imagine how delighted we are with the continued progress of all of that there. I have to say again, that just the consistency of this functional data shared with Dyne, particularly impressive. And the combination of dystrophin levels and functional improvements is what the community are looking for whenever they're making decisions. So congratulations, team. That's a result that the community wanted to see. We're looking forward to further updates as the company prepares to file their BLA for accelerated approval very soon in the second quarter of next year. A post-webinar email will be sent tomorrow should you have any additional questions, and we'll get in touch with all attendees to let you know when the webinar is posted online for sharing. So for everybody, thank you everyone, particularly Dyne, but all of the ones that joined us today, and we really appreciate it. Have a great rest of your day.
Michael, let me just thank you and Liana and Deborah, early supporters, great partnership with Dyne, very, very important to us. And to all the patients and families who have sacrificed
Yeah.
to come into this clinical trial, we appreciate it and thank you.
Thank you too, Doug. Much appreciated. Thanks for those words.
Bye, everybody.
Everybody.